Hello everyone, very good evening. Please confirm if I'm audible to you. Yes, very good evening. Pradeep Mukherjee. Yes, Nidhi, Dikeshwar. Thank you so much all for joining. Very good evening to all of you. Yeah, so who else do we have over here today? Achal, welcome all of you to the session. Abdul, Paras, wonderful. Vagueleaf, Neelam, Harshangi, Anuj. Yeah, so a very warm welcome to all of you. And a very good evening to all the participants who have joined us today. Thank you for joining us. And we are about to begin our session now. So I hope you are ready with your notes and your note copy and your pen because I can guarantee there are a lot of takeaways that you're going to get through this session. So let's get started. Yes, I hope you all are ready, right? Wonderful. So I'm Urmimala, your host for this session, and I'm very excited to see so many of you here for our webinar today on integrating AI ML into your bioinformatics and biology research. We'll learn about both platforms and pipelines. Okay. So you must be wondering why are we choosing this topic today, right? Out of so many things and so many topics that Biotechnica conducts a webinar for, because the reason why we have chosen this particular topic today, let's be very honest, you have been hearing the words artificial intelligence and machine learning for a long time now. But I can say they are no longer buzzwords. Just they are not just buzzwords that some fancy terms because they are already reshaping modern biosciences. Think about it. 10 years ago, predicting protein structures took months of computation. Today, with artificial intelligence tools like AlphaFold, researchers can get near instant models that is already revolutionizing the drug designs. Same happens with whether it's for CRISPR optimization, gene prediction, or even NGS data classification. So things that once felt impossible, they are now everyday research tasks. This shifts means one thing for all of us here. If you are serious about building a research career in biology or bioinformatics, practical AI and ML experience is no longer optional for you. It is essential. Okay. And since Biotechnica brings you the hottest trends in town, so that's exactly what this session is about. Over the next 1 hour, our experts will guide you step by step from understanding what AI/ML actually means in bioscience to learning the right platforms and tools, and finally, how to build practical pipelines and projects you can showcase in your career. But before having our speakers here, before introducing them, I would like to remind you, all of you, this is important, so please pay attention. At the end of the session, you're going to get a free gift from us. Yes. So those who will stay till the end, you will get a free gift from us, a freebie. But if you are interested in getting the freebie, not only you must stay till the end, but you also have to register in this link. Okay. So the link is there already in your comment section. You can check. So you have to register yourself in the link if you want to get this freebie. Okay. Then only you are entitled to get it. All right. Yeah. So everyone got the free link. Everyone got the registration link. Yes. So you can register over there. Don't delay right now. You click on it and you can register over there so that you all get your free gift at the end. And obviously, you have to stay till the end as well. Okay. So now let us quickly welcome our speakers for the day. And this is how we will have the flow also for today. First up, we have our very own in-house Dr. Neeraj Kumar. I welcome you, sir. So as you all might be familiar, yes. So as you all might be familiar with Dr. Kumar already, he is a computational biologist who uses artificial intelligence to speed up drug discovery. He earned his PhD in bioinformatics from CSIR IHBT and ACSIR, and then completed postdoctoral research at Pennsylvania State University, where he built algorithms for antibody analysis. Dr. Kumar is skilled in tools like TensorFlow, PyTorch, RDKit, and GROMACS, and he has published in leading journals such as Journal of Cheminformatics and Medicinal Research Reviews. He has also cleared competitive exams like UGC NET, GPAT, NIPER, JEE, and GATE. Today, Sir will walk you through the what career roles are there, what actually AI/ML is, and how it is relevant to your particular life sciences field. Okay. Then we have Dr. Anuprabha with us. I welcome you, ma'am. Welcome to the session, Dr. Anuprabha, ma'am. Hello, ma'am. So Dr. Prabha is a project product manager at Illumina, where she leads the development of next generation genomic solutions that integrate advanced analytics, scalable platforms, and AI-driven insights to transform precision medicine and clinical diagnostics. With over a decade of experience spanning genomics, bioinformatics, and healthcare technology, Dr. Anu has previously driven product innovation at Haystack Analytics, delivering automated whole exome sequencing, pharmacogenomics, and preventive healthcare platforms with industry leading turnaround times. Dr. Prabha holds a PhD in computational biology from IIT Bombay and has a proven track record of bridging cutting edge science with user centric product design to accelerate diagnostics and also improve patient outcomes. It is great to have you, ma'am, today with us. Thank you for joining.
Thank you.
Finally, we will be joined by Ms. Snigdha Tiwari. You all will be knowing, ma'am, if you are part of our courses. Yes, so Snigdha Tiwari, ma'am, is a computational biologist and clinical research specialist at the Biotechnica CRO. She recently completed her PhD research at IIT Roorkee, where she worked extensively in computational biology, protein ligand interactions, molecular docking, and clinical bioinformatics. One of our key achievements was developing the Utsarjan app, a digital health platform for pediatric nephrotic syndrome in collaboration with AIMS Delhi. With over 5 years of experience in bioinformatics, telemedicine, and healthcare data management, she bridges strong academic training with real world translational bioinformatics. Thank you, ma'am, for joining us today, and we're looking forward to your session. So, are you all ready? Yes, so we will be having the Q&A session at the end. We have a live Q&A where all our eminent speakers for the day, experts in their field, they are going to address your questions. So, make sure you stay with us till the end to get all your doubts answered directly from our experts. Yes, thank you so much, Ritesh. I'm sure it will be fascinating. So, without any much further ado, let's get started with the session. I would now like to hand over the mic to our first speaker, Dr. Neeraj Kumar. Welcome, sir. Please take over the.
Sure, ma'am. So, good evening, everyone. I think my slides are visible. My screen is visible and I am audible to you.
Yes, sir, it's visible and you're audible too.
Okay. So, let's get started. So, today I am going to speak over uh, making sense of artificial intelligence as well as machine learning for life sciences, life science students and researchers. So I am Dr. Neeraj, and I would like to thank Dr. Urmi for introducing me to you or to all of you. So let's get started and dive deep into the domain which is called AI and ML. So AI stands for artificial intelligence as well as machine learn. ML stands for machine learning. So what is specifically machine learning and artificial intelligence? So AI is the broad field of creating machines that can mimic human intelligence, right? So machine learning is a subfield of AI. So here you can clearly see that. So this is artificial intelligence. So the machine learning comes under the umbrella of artificial intelligence. So what is artificial intelligence? So artificial intelligence can simply be explainable that it is creating intelligent machines that is mimicking human intelligence, right? Where machine learning are the collection of algorithms, okay, so which are making the machines intelligence artificially. That is why it is artificial intelligence. So what type of machine learning algorithms or the other algorithms which are making machines more intelligent. So these are neural networks and deep learning. Okay, so what is neural networks? So neural networks are computational models with interconnected artificial neurons. Okay, so that is where they tries to, or they start mimicking the human brain. Okay, so neural networks are a collection of networks or models which are connected together to process the information, to learn from that data or the information, to understand the patterns, and on the basis of them they tries to predict the details of the data. Okay, so underneath the neural networks there are deep learning. So you must all of you are, you may be aware of the deep learning these days, because it is the buzz of the town and talk of the town. So when there are a collection of various or diverse neural networks to process the complicated data or the complex data, or to understand those complicated or the hidden patterns, there comes the action of deep learning. Okay, so from here you can simply understand that neural networks with multiple layers for hierarchical representational learning. Okay, so the collection of these algorithms and the foundational algorithms of machine learning tries to make the machines intelligent artificially. That is why they are called, or the term is called, artificial intelligence. Okay, so artificial intelligence, simply the machine that mimics human intelligence. Further, I just simply explain each and everything regarding AI as well as ML. Okay, so what are the basic examples of AI as well as ML in biology? Okay, so if you want to just predict the disease outbreak, okay, so you just need to have the patient's data, and then you can train your AI algorithm, and on the basis of that it will learn the patterns of the data, and it will try to predict that when would be the trends are showing, or the pattern of the data showing that when does a particular disease can outbreak. Okay, whereas the ML algorithms can simply classify the images from various medical data sets, like MRI scans, CT scans, X-ray scans, etc. Okay, so on the basis of that, the AI algorithms can understand or can learn the pattern, that what type of disease is coming out and what type of outbreak can happen. Okay, so that is the basic function as well as basic applicability of AI and ML. And what is the catch? The catch is they can understand the data. They can understand a larger set of data, the humongous amount of data in a shorter span of time, because we have the most advanced computational architectures these days. We have GPUs, we have TPUs. A TPU stands for tensor processing units, etc. So that is the artificial intelligence, and that is the machine learning, and that is the power of machine learning. And in the world of data, we need to understand these basic things, right? So I think you might have the basic idea about AI and ML, and why it is needed in these days, and why you should have a skill of AI and ML. So what are the roles of AI or ML in life sciences? So first of all, the pattern recognition. Okay, so AI has a ability to recognize patterns in crucial in life sciences, as it can sift through huge data sets to find relationships that would be invisible to human eyes. Okay, for example, in genomics it tries to identify the genetic patterns linked to the disease. Okay, and in drug discovery, they tries to find out the new molecules as well as the compounds that could be effective drug candidates for a specific target under study, right? Then machine learning can be used for automation. Okay, so AI powered automation takes over repetitive as well as time consuming tasks. Okay, which is freeing up the scientists as well as the clinicians to focus on more complicated work. Okay, so let's just understand it by example. So AI is being applicable being is is being applying for lab automation. So what is lab automation? It is where the robots powered by AI can perform experiment, can handle samples, as well as run tests with high precision as well as speed. So it is a process known as high throughput screening. Okay. Further, the automation can also be implemented into data entry and analysis, where AI can automatically extract and process the information from research paper as well as the electronic health records, which can eliminate the manual data entry errors as well as speeding up the literature reviews. Okay. Further, AI ML can be implemented for or can produce predictions. Okay, so by means of predictions, it can predict the future outcomes on the basis of the learned data or the past data or the events happened in past. Okay, so from data records, that is the influx of data or the inflow of data used to train the model, can help you to learn the pattern as well as to understand the data as well as can make predictions for future outcomes. For example, it can be used to predict clinical trial success. Okay, so by means of clinical trial success, whenever we have a potential drug molecule, it needs to be undergo through preclinical as well as clinical trials. So it should be tested over a sample of few patients. So if we find out some positive responses, then we can say that this drug can be applicable over the larger data sets or the larger people, or can be implemented generically. Okay, so here AI can analyze data from past trials to predict the likelihood of a new drug succeeding, that can save billions of dollars as well as years of research. Okay, so it AI can design the clinical trial test, can assess the outcomes, as well as can guide to make a more what you say, the successful candidate that can pass through the clinical trials, right? So that, that is the process where AI is humongously accelerating the drug discovery process, right? Then AI can be used for data integration. Okay, so data in life sciences is often fragmented, and it is stored in different formats across multiple systems. For example, electronic health records, lab records, the lab results, research databases, etc. So AI is key to integrating all the diverse data formats. Okay, so for example, it can be used for unifying data sources. So AI can connect and standardize data from various sources that can create a single and as well as the comprehensive view. Okay, so the unification of data sources can allow researchers and doctors to access a complete picture of a patient's health as well as disease characteristics. Okay, so that these are the basic applications, or these are the revolutionary applications which is tremendously accelerating the discovery process, understanding process, and the innovation process, right? So, what are the basic types of machine learning? So, machine learning can be divided into various types, but the major types are unsupervised learning, reinforcement learning, supervised learning. Okay, so by means of supervised learning, so here the data set is labeled. So data set comes with the categories or the details about about the samples, about the patients, or the health record details, etc. So where the data set has the category and can comes with the label. So the application of machine learning algorithm is called supervised learning. So it can be classified into classification as well as regression. So it has two types. The supervised learning has two types: classification and regression. And these are the basic application where the classification is applicable and where the regressions are applicable. Okay, the second type is unsupervised learning. So here the data does not have any kind of what you say, the labels or the categories or any kind of details, the generic details on which we can understand that what type of data it is and what it is carrying exactly. So the data which is not categorized, not labeled, So we need to understand the data, we need to find out the patterns, we need to find out that what type of trend the data brings in. Okay, so there comes the unsupervised learning. Here there is no supervision over the data sets. However, we tries to find out that what are the patterns, and on the basis of those patterns we tries to cluster the data. We just put the data set in various classes, and we try to categorize those data sets. So on the basis of them, we have two types of unsupervised learning, that is clustering as well as dimensionality reduction. So these are the applications of the clustering as well as the dimensionality reduction. So you just can go through them. Okay, the third part of the machine learning is reinforcement learning. So whenever we have the regular flow of data, so it is a continuous process. Okay, so whenever you are playing a video game, okay, and the person who is playing the video game and interacting with the video game, the character within the video game is tries to assess the data set. Okay, so you tries to input the data from the remote control, or the remote control or the console. Okay, on the basis of them, the character within the game tries to improve its skills on the basis of user input. That is called the reinforcement learning. Okay, so here the algorithm tries to improve itself by continuous influx of data. Okay, so these are the various example of the reinforcement learning. So these are the basic type of machine learning. So this is just an overview. So you need to dive deep into the regime of the machine learning to understand in depth, right? So the next is there are various machine learning algorithms which falls under the classification reinforcement learning. Sorry, not classification directly, in supervised learning, unsupervised learning, as well as the reinforcement learning. Okay, so here the support vector machine. So this is the first algorithm which falls under the supervised learning category. So it can be used for both classification as well as regression. Here the k means it is an unsupervised learning algorithm. The logistic regression and linear regression and random forest, these all falls in the in basically in the supervised learning, and can be carried out for regression as well as classification tasks. Okay, here the naive bayes, it this basically it is a mathematical theorem which drives the whole the machine learning algorithms. It can be used to improve supervised, unsupervised, as well as reinforcement learning. Okay, then we have the k nearest neighbor algorithm. Okay, which falls under unsupervised learning, which is basically used for clustering or dimensionality reduction, etc. So these are the basic algorithms, and if you wanted to understand these, you need to understand these more, how they are, what are their basic principles, how they are applicable, how they are being designed, and how they are being implemented into the healthcare or the biomedical science or the biotechnology. Then you need to you can come to Biotechnica, where we are teaching all about these things, as well as we are having various range of projects, where you can join the particular project, as well as you can have the hands-on experience, as well as the further details, etc., and you will be future ready with the new skills. So what are the real world applications? So as Urmi ma'am just talked about or discussed about the AlphaFold. Okay, so AlphaFold is a talk of town from last 2 to 3 years. Okay, so what is AlphaFold? AlphaFold is a algorithm which is used to model protein structures from protein sequence. Okay, so you have a sequence. You do not have its X-ray structure, or the structure resolved by a cryo-electron microscopy or NMR. Okay, so this is an algorithm which automatically builds the protein structure and produce a 3D structure. So how it exactly work? So let me just give you an overview that how it exactly works. So first of all, the AlphaFold, the algorithm of AlphaFold takes input as amino acid sequence. That is the protein sequence. Okay, that is a polypeptide chain, not structure. That is just a chain. So what it tries to do, it tries to learn the feature. So that is called feature engineering. Okay, and here the tools which are being used called pairing and transformations. So that has been carried out by the transformers. So transformers comes under deep learning. Okay, those are generative algorithms. Okay, or they are variational autoencoders. Okay, so that is how the feature engineering is being carried out. Further, these features are being extracted and optimized, and then a particular model is being pre-trained over those features, and then these pre-trained models tries to build the structure. Then further, that is called the structure relaxation. Here the molecular simulation is being carried out or run out to optimize the structure, and then it produces the 3D structure of a particular protein sequence, that is the amino acid sequence. So we have just given ATC, sorry, not ATC, the one letter codes of amino acids. Okay, so now we have the 3D structure. So that is how the AlphaFold works. So what technology they have used? They have basically used the deep learning. In deep learning, they have used transformers. They have used autoencoders. They have used convolutional networks. Okay, they have used artificial neural networks. They have used the basic foundational machine learning algorithms. Okay, so that is how the deep learning or the machine learning or the artificial intelligence is changing the health domain. And uh, uh, this is the high time to learn new skills to be the industry ready or the research ready for our future. With this, I would like to thank you and giving me the opportunity to talk and to discuss with you guys. Thank you so much, ma'am.
Thank you so much, Dr. Neeraj, for simplifying AI/ML in biosciences and showing us its real impact. That was
Thank you.
truly eye-opening, and our audience understood what AI/ML really means for life science research and about what algorithms are involved and real-world success stories that are happening all around. So thank you once again, sir,
Thank you.
and we will have you back during the Q&A session as well.
Sure, ma'am. Sure.
So, with that, we now move on to our second session. I welcome Dr. Anuprabha ma'am to kindly present the screen and take over the controls. Over to you, ma'am.
Okay, thank you, Dr. Avni and Dr. Neeraj for setting up the platform for the next talk sessions. Give me a minute to just, yes. Is my screen visible? Am I audible
Uh,
clearly?
yes, ma'am. Audible and your screen is visible too.
Oh, great. Hi, everyone. I'm Anu, and I'm currently Technical Product Manager at Illumina. I have been a product manager for close to 4 years now uh, in genomics, mostly uh, whole exome, whole genome analysis. And I would like you to take you through some of the product pipelines that we do, how product comes to fruition, what is productization, and all that stuff. So let's move on. Okay. So before we move on to what we want to do, we should also know where we want to do this, right? Now, why we would want to choose a specific kind of platform for our development environment, or any sort of pipeline deployment or whatever it is. Now, why this platform matters a lot? Why is it important? Because the platform will dictate uh, if you could handle large data sets, small data sets, whether there a possibility of reproducibility. Can we test the environment? Can we test the outcome? Can we see? Can that pipeline or product be scalable across, thousands of samples, tens of thousands, depending on, what kind of product you are developing. Also, it can also help you in creating a proper versioning system that can help you trace your steps back in case something goes wrong. It will also help you understand what modules for me are working or not working of the pipeline, how to do troubleshooting, so on and so forth. This will also create a very good platform for you to showcase the validity of your results when you are trying to apply for, sort of some regulatory bodies. And it also keeps track of all your documentation needs, which is very, very important when you want to have a product in the market. Okay. So understanding what are the platforms that we would use in the product journey. Now there are three different kind of platforms that we use. First one is there are scripting platforms. These are sort of platforms and tools that you use when you want to write the script, what you want to do in the product, what is the logic when you are building it. So you need an environment where you can actually code for it, basically. Now, the second kind of platform, the kind of platforms we're talking about are modeling platforms. Now, once you have, sort of used, have taken data, cleaned it, you have transformed it, you have loaded it. Now you want to do something with your data. Now, when you want to do something with your data, maybe you are trying to do some ML exercises, if there is a classification problem, I want to do some classification. So I need to understand, now that I have all my data loaded onto the system, what kind of model I want to build, what is the most optimum model, which will give me the best results, how will I validate that? All that you can do on modeling platforms. Now, these are where you are, again, basically write your code, but also, sort of see outcomes and, sort of see outcomes, graphs, any sort of tests you want to do, any sort of validations you want to do, so on and so forth. Now, all of this makes a lot of sense when I'm working alone. Okay, I have my system, I am doing something, I have a problem in mind, I have written a very good script of it. I feel at the end of it, to make it usable, I have also made some ML model. It is helping me in some problem that I'm dealing with. But will this entire bit of product, whatever I have developed, will I be able to use it on your system or anyone else's system? Probably no, because it will have many issues if I want to run it on your system or any other system. So the system to system variability is something that is a biggest problem that people face, especially when they are working, when they are creating a SaaS product. So what to do there? I want the efficiency to be right. I want the code to run flawlessly. I want it to not have much lag. I want, sort of parallel processing. I want all those dependencies solved. So what I do, I have some deployment platforms in which I can run those. So they will take care of, obviously, my time, speed, requirements. Also, they will take care of all my dependencies, because I have some containerizing applications like Docker. And it will also help, my multiple web services to be run back to back. So now, given that outcome, now having scripting platforms, modeling, and deployment platforms together is what will enable me to run the entire product from end to end. Okay. So I have just put this. I have, like, long back used some of these, but then we moved to better platforms. But these are some of the beginner friendly platforms that I would like to bring your attention to, because it doesn't. Why these are very important for you to try it out for yourself if you are a beginner, because it doesn't take a lot of time to set up. You can quickly, do some experiments, run some real data, and sort of design an experiment and actually go through the entire process of what I want to do. Am I able to do it or not? Am I able to create a model? Am I able to validate that model? So on and so forth. um, it is also very interactive. So generally, when you do in product development, the entirety of the code is run. And if you want to, if you are a beginner, you would want to see whether each small step is running correctly or not. So it gives you visualization therein in the interface itself. So it's easier for you to run through your code sort of line by line. Yeah, and it just has a ton and tons of modules, pre-made modules, which are there for your perusal, for which you can use as much as possible. And there is tons of there is a big community out there to help you out with this scientific community. If you have any problem, if you are facing some errors, or if you are trying to troubleshoot something, you can always go Google it, and you will most probably have 50, 60 answers, or I do not know, 500. Depends on the problem, but you will have a repertoire of scenarios in which people have used that, and they have found problems and how they have solved it. And it helps you a lot to start with a problem, at least, if not anything else. Okay, so how does it a pipeline looks like mostly? So um, what a real pipeline is, maybe lines and lines of code, but there are different stages of it. So generally, the initial part of it is, what data are you going to use? Now, once you have that data, how you are going to extract that data, how you are going to clean that data. Does that data need any sort of transformation? Does it need to be in a specific format to load into your pre-existing system that you have designed? So basis all of this, you can go through a multitude of steps, depending whether you're working with genomic data, you're working with proteomic data, you're working with even structured data, depending on what is your input. The next step is feature engineering. Now, if you are building specifically an ML model, you need to understand what feature is going to impact the outcome. Is it positively correlated? Is it negatively correlated? Or the feature has no role at all in the outcome? In case that is, you try to optimize your feature set so that it gives you the best possible outcome. If a feature is not influencing your outcome whatsoever, and it is not per se sort of a tag or anything that you want to track through the entire process, it would be very wise to actually leave it and only create models using features that are usable or have some influence on the outcome. The next step comes model training and validation. Once you have actually made the feature set that these are the variables I need to account for to predict my outcome, now you need to train your model. Now, very once you train your model, then you can test it on a test set. Using your training and test set in different combinations, you can do a lot of validation. There is different kind of validation methods out there. You can go ahead, Google it now, and you'll have so many kinds that will pop up, and you can use any one of them. What is important is not how many methods of validation you know. What is more important is, can I use which method of validation will suit my experiment the most? What do I want to assess? Okay. The last one is results interpretation. Once you have made a model, have seen the outcome, you have tested it, now you have validated it, now this is the kind of outcome I'm looking out for. The only value of it is when you translate it into sort of biological context or a biological real case scenario in which you are going to use it. Otherwise, it is just an exercise, a research exercise that would maybe help in understanding something, but will not be, you know, in terms of applicability, might not work as much. So even though these terms I know does excite everyone, yeah, like these are the tools and everything, it does excite everyone a lot. But I think the emphasis should shift from what the tools are there, or what are the different kinds of methods that are there, from there to actually understanding your experiment better, understanding what you want to find, understanding what I need to use to get that outcome, what is the outcome I'm chasing, and what I want to, sort of validate it with. What are the case scenarios in which I can understand whether the outcome is true or not? What will be a true positive for me? What will be a true negative for me? All this understanding is more important than, what kind of ML model you use or what kind of AI you want to do, to be honest, because any ML model you use, any kind of AI you try to make, it will always have an output. Understanding that output and putting it in context of the experiment is what is going to tell you whether it is, usable or not. If I can do the same thing without AI, I'll probably save a lot of money in, running it through EC2 instance, and having it using so much GPUs and all that stuff. So I would rather focus more on what experiment I want to design, what kind of validation and case scenarios in which I'm going to use, and what will be my true positive, true negative, false negative. Accordingly, I can, assess what is the accuracy of my model, etc. You know, there are many such questions that holds more value than other things, what I have mentioned. Okay, coming to languages, libraries that are there. So you can either use Python or you can use R. However, in my experience, industry standard requires Python. R is phenomenal for research purposes, and especially in some problems in the transcriptomic world, it is very important, and people do use it a lot. But is because every other pipeline platforms actually prefers Python. The usability is much more conducive to the Python environment, whereby people are trying to change from different environments to Python. But if you are starting out, it is more important, as I said earlier, to focus on the concept, focus on the experiment. I would strongly suggest please use R and try to, run through it and see, even in R, you can very easily see the outcome then and there. You can plot graphs and see whether it is working for you or not, and the outcomes, and you can try and analyzing in any way you want. Okay, so coming to essential programming tools for bioinformatics. Uh, Now, these four are very important aspects of product. If you think about it, there is no product if these concepts are not in place. So, version control, the every time a product is made, there are going to be changes in product, there are going to be changes in, maybe data sources that you are working with, and all these will have different versions. If you have gone to any database, public database out there, or any publicly available tool that is out there in genomics or otherwise, they always mention version, because the outcome may or may not change uh, depending on the version. And if you take it in the context of databases, even when you're writing a research paper, or when you are, submitting a paper, you have to write the versions of databases that you have used. If I have used, for example, the genes database today, and I use it after, 2 to 3 long years later, the outcome will definitely be different. So that is why having some sort of version and version control is very important. Git and GitHub is an excellent way to track changes. It is also excellent when there are generally in industry many people work on the same problem. So it is very important platform for all of us to collaborate, to manage the product, and it has a lot of sort of utility in tracking issues, problems, bugs, feature you want to introduce, so on and so forth. And if you have time, I would really encourage you to go and see the GitHub tree, what it means, what is branching, and all that. It is really interesting and will very much be helpful for you. Besides that, there are package managers. So basically, when you are trying to run a pipeline, you want it to be run in a certain environment. Now, we want to create an environment so that all variables are accounted for, all dependencies are accounted for. For example, I have made a pipeline which intake needs 15 libraries from 15 Python libraries, 10 Bash libraries, 10 another R libraries, or some Java classes or objects or whatever it is. So what I would want to do is have an environment that encompasses all of these dependencies. So whenever I run the code, I don't need to run it again and again. So all the backend dependencies that are required to run that code, to run that entire pipeline, are managed by these managers. So basically, they will create an environment in which whatever you install, that will be sort of global variables for that, and you can repeatedly use them, and you can export to the environment. You can import to a small module. It is amazing how you can use that. Besides that, you have workflow managers. Now, workflow managers tells you basically your pipeline is going to be a list of many modules, many codes, and they may be written in different languages. So you can use a Bash wrapper, you can use SneakMake, Nextflow. So all of these, how does it help? It runs your different modules, different codes, or different aspects of your steps in your pipeline one by one automatically. Now, if you want to come to a desired outcome, there will be many steps. Now, all these steps will be doing multiple processes. Now, you want to run everything. Now, you want to also have them run one by one, because not necessarily one by one, depending on the problem, but many modules that you would have the output of one module would become the input to the next module, so on and so forth. Now, this small modularized development is very important to track changes, to do unit tests, for testing, for doing integration testing, and it helps a lot to see the sanity of the code and to check the sanity of the outcome. What also it helps is it also creates a lot of logs. So you can have time logs, you can have bug logs, you can have a lot of them. If there is an error, there will be it can catch errors and create logs. So all of this will help you in troubleshooting to know, out of these 5,000 or 10,000 lines of code, where I might have gone wrong at which place, and it helps you a lot in troubleshooting. Now, all of this makes great sense till you are lucky, working on your local system. But the moment you have created a SaaS, or created a web interface, or created a service, now you want to run this simultaneously on many systems. On one system might be at a very different place, another system might be a very different place, and everyone would be just opening that web page and trying to run an instance on that software. So on that service. So what do? You do containerization. So basically, Docker is a tool that helps you do that. So what it will do, it will wrap around your environment, and it will take care of all the dependencies and everything inside it. So whenever you run it anywhere, it will first install all the possible dependencies that are there, export all variables, create all the folder structures, or anything, any outcome that you want to get out of this. It will create all the folder structures, from where to take an input, how to capture input, how to give out the output, all that stuff. So in clinical scenario, when you are creating a service or you are creating a product and you want it to be run by people around so that they can use that service to solve a problem in the clinical world, this is something that is absolutely must. Okay. So how to pick the right tool? So as I said, there are millions and millions of such tools. There are millions of such services, and there people every day that we are sitting and talking. There is someone who has come up with some problem they have faced in their real life scenario while developing a product, or while developing or using these tools. They have come up with better options. Better alternate options that they have created, and all of these are there for the public to use. However, it should be not the one guiding you what to use. The latest trend is not necessarily what to use, but I think the more dearth in the industry is identifying that what is the problem and how will I be able to solve it, and then go into what are the tools I can implement in such scenarios. So if in case I have a biological problem, if I have a clinical problem that I want to solve for the market, you need to sort of delineate into smaller paths, smaller bits of development. What are the aspects that I will develop first, what are the in what order will it be developed, and accordingly pick a tool for each one of them. If I can sit here and I can break down the experiment into logical steps, and I'm aware of what kind of outcome I'm chasing, the tools is only going to play a support part. It is only going to enable you to do the right things with the right sort of toolkit with you. Okay. Again, so matching tools to your data type and skill level, depending on the kind of skill that you have. If you are a beginner, if you're just starting out with this, or if you have already been in this, done some experiments, and you want to try out, um, uh, uh, some intermediary level tools and see how it goes, or there is if your skill is at advanced level where you know what products are, what pipelines are, and you want to work with them. I've just put some examples here for you to try out, and at least for the beginner and intermediate is something that you can very well just go out and try. Jupyter with BioPython is like just an interface. You just write it on Google, you get an interface, write just hello world, I don't know, just print some lines, go ahead, do small pandas operations, go and check out for yourself. Similarly, for our studio with Bioconductor, it will require some downloads. Similar with Nine, there will be few downloads, and you will need to update some of its libraries before you can use it. But both, again, Nine, if you have a logic in mind, you already know how to solve a problem, you can build it using small nodes. So they have nodes which you can stitch in a logical way and run and see the outcome immediately. It is very interesting tool to help you think and solve problems. Once you know how to do all of these, then going to building custom Python pipelines, making bash wrappers, using Nextflow, SnakeMake, and all that makes sense. Okay. So these are some of the things that I have literally just picked up from the web. That these are hands-on learning resources. Basically, if you go there and you can Google any of these, or you can find more by yourself. Just take any problem for which people have done something, and so that you are on the right track, and try few out by yourself on the simplest interface you can understand. Learn how to solve problems first than learning how to solve tool first. Once you solve the problem, you'll get the hang of any tool, because now you know what are the logical steps that is important to solve a problem. And for each logical step, you can find the relevant tool, see the pros and cons, and accordingly use depending on the problem that you are working through. Oh, so this is my last slide. So thank you, everyone. It was great interacting, great presenting to you guys. And over to you, Dr. Umi.
Am I audible now?
Yes, yes, you are.
Yeah, thank you. Thank you so much. Yeah, I guess there was some issue on my end. So thank you so much, Dr. Anu, for that practical deep dive into platforms and tools. I'm sure our audience now knows exactly where to begin experimenting. The way you talked us through the must-know platforms and libraries, from beginner-friendly tools like Jupyter and Google Colab to advanced libraries in Python and R, and explained how to choose the right one, was truly amazing. So thank you so much, ma'am, for your time, and we would request you to please address our students' queries during the Q&A session as well. We will carry on now with our third session. Yeah,
Thank you.
thank you, ma'am. So yeah, so let's quickly jump on to our third session. I request Ms. Snigdha ma'am to kindly present the screen and take over the controls for the third session. Over to you, Snigdha ma'am.
Thank you. Thank you, Urmimala ma'am. Now that everyone has gained a good overview on AI and ML, very good lectures, of course, by Neeraj sir and Anu ma'am. I will quickly share my slide. Just a minute. Okay. Yeah. Getting a little. Okay. My screen is visible, ma'am?
Not yet,
No?
Snigdha ma'am. Yeah, now it is visible.
Now it is visible. Okay, okay.
No, it
Uh,
went back again. I guess, yeah,
uh,
the PPT. Yes.
the PPT is visible now, ma'am?
Yeah.
Okay, 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 fine.
Okay.
So now hi everyone. I'll extend my lecture on how to learn and build practical pipelines for research. Okay. So guys, learning and AI and ML and executing mini research projects in biology and bioinformatics with AI and ML. I will also let you know the practical pipeline for research. You know how to get the practical guide for it. Already Neeraj sir and Anuprabha ma'am very nicely told that what are the basic pipelines, how to just start your basic projects with Jupyter, with Python libraries. You can go ahead, you can take up any analysis, any troubleshooting any disease, any of the data, and then you can go ahead explore from basics of pandas, numpy to scikit-learn, tensorflow. So actually, why hands-on project are so essential? um, so why hands-on is so essential is actually for developing a portfolio. Theory is not enough. Academic courses teach foundational concepts, of course, but they don't actually prepare you for real world data challenges, right? Projects actually build this gap, the gap between what you learn, what your concepts have been built. Taking up a project and doing the pipeline development shows the potential, and actually it only bridges for the real world data challenges. Coming up towards the development of a practical skill, project force you to master tools, troubleshoot your errors, and manage your complicated data sets. Working with ML, big data, big, because we are in the world of high throughput, ML, AI, big data sets, complex data sets, of course, because we are working with biology, genomics, omics, drug structures. Developing a practical skill is a necessity. Also, building a portfolio is very important. A strong portfolio, guys, of the projects is valuable, for the employers and admission committees, and there are the list of course names. So just writing the list of course names and what academic courses and points and everything you had, more than that, now building a portfolio, what is your practical enhancement, what are your practical skills are more important, towards building. And of course, whoever is listening me, for transforming the careers in the industry, has to do that, right? The advantages of a dry lab projects, picking up a dry lab project, what are the advantages that one gets, with the dry lab. So talking about what is actually a dry lab project. So the project that relies on the computational analysis, right, for existing data, maybe you've picked up the data. Ma'am already told that you can pick up the data from different databases. There is ncbi, there are different kind of boom of databases, of course we have. So without needing a physical wet lab experiments, you can just pick up the data and then start your pipeline development, like end-to-end development for your analysis, pre-processing, and then working with the ml data sets, supervised and unsupervised learning, all that. um, Accessibility, talking about the dry lab projects, that can be done anywhere with the computer, the internet, a good internet connection, your storage value should be good, computer ram, and your internet connection should be good, and that is pretty much goes up with the dry lab experiments, making your projects, making them ideal for students and professionals, of course, without access of physical labs, right? They are cost effective as well. They require minimal finance investments, unlike wet lab experiments, which might be expensive a lot of times. All the chemicals, overhead expenses, all of that. Time is one of a very good deal here. Actually, they can be completed in a relatively short time frames, because of course, compared to long time wet lab studies, if you see, there is a time enhancement, and we get a time frame benefits of with the dry lab projects and pipelines. Planning a project with a research cycle. uh, there's a whole of a research cycle with picking up any of a project. uh, the data collection is your first should be your first target. Picking up anything, because of course you have to analyze your data, right? Once you have analyzed that, this is my research gap. Once you have chosen your research gap, picking up a data is the first and the foremost thing that you'll do. um, Going ahead, after the data collection, you will want to develop your model. How will you develop a model? Already Anuprabha ma'am talked about it. Whether it will be a supervised learning, unsupervised learning, you have to choose according to your endpoints. What is your hypothesis, what are your endpoints, what are your protocol development. Based on that, the model development, of course, you're going to do. After the model development, one has to do the evaluation. What are your outcomes? Those outcomes, what will be the outcome detail, what will be the evaluation, right? After you have the data with you. The problem identification, no, sorry, I'm really sorry, starting with the problem identification. Okay. So starting with the problem identification, and then data collection, and then model development, and then at the end you will of course want to do the evaluation, right? You want to analyze your result. So this is how the planning your project should be, ideally. So first, go with defining a problem. Start with clear, specific, and answerable research questions. Say for example, you want to what niche you want to work with. What is your research gap that you found with your literature review? That is important. Then after that, you'll do your literature review, you'll research what others have already done. um, in the previous lectures, you have already got a good insight of start picking up what has already been done, so that that I'm with the good path, I'm at the correct path. So identify the gaps of knowledge. After doing the literature review, you have to collect your data for the analysis. So collect your data, find find what is there, acquire publicly available databases, what is relevant with your problem. whether working with drugs, omics data, whatever be. After your data collection, one should analyze what is the methodology. Plan your analytical steps from data processing to developing a pipeline, then training, and then the evaluation. Execute and analyze after you do the methodology, after you develop your protocol, after you develop your pipelines, you know your endpoints, you know your objectives, you go ahead and do the execution as start with the Jupyter, start with the basic pandas, numpy, basic of all the Python. Maybe you can pick up R. Ma'am told it is a very powerful for statistical analysis, a very powerful tool, and that is very, that is true. R actually gives you a good insight. Start with the basic ggplot and all of that. Execute and analyze, run your code, generate result and interpret them. At the end, try to present, right? The presentation is important. Why try to present is, you also want to communicate your findings. You should communicate your findings in a clear and compelling way. Example, what report have you presented, what report have you made. You should be able to convince the other, anyone, any scientific community or anyone for that, right? So that is a part of a cycle, a part of a research cycle. Talking about step one, defining your research problem should be between a good problem and versus a bad problem. So a bad problem will look like, I want to cure a cancer with AI. So this will something a bad problem will look like. With this, what I meant is, I want to cure cancer with AI is too broad, right? What you want to study, AI is just the application of ML. So what ML pipelines you are going to apply, what generations you are going to do with AI. So this is a bad concept. The good concept that you should, I highly encourage you to go with a good concept. The good concept can be, uh, what can a machine learning model predict the drug sensitivity of maybe of breast cancer cell lines based on their gene expression profiles. So based on this problem, your research cycle, what machine learning algorithms I want to pick up, what databases I want to pick up. I want to go to genome expression omnibus or TCGA, or those of lines of databases. So your machine learning, you want to be explore the cluster algorithm, regression, classification. So this is the good problem. So one should pick up, for a research cycle, one should pick up a good problem, one should know. Step two is collecting the public database. Once you know your research gap, you should definitely, of course, you have to collect the data, right? So the bioinformatics community has already generated a massive amount of high quality data, and we know that fair use is all, always read and adhere to the usage policies of the data, whatever be the data repositories. um NCBI, GEO, RCSB, PDB, and TCGA are a few of the public databases that I've just picked up. So NCBI, or Gene Expression Omnibus, is the primary sources of gene expression databases, guys. RCSB, PDB, if anyone wants to work with big data, like the big biomolecular molecular dynamic simulations, talking, MDS analysis. So RCSB, PDB, UniProt are the main archives for 3D protein structures, right? Uh, Talking about the TCGA, this is a rich data set for genomics, um, cancer clinical data for various cancers. There are a lot of clinical data that is available for oncological developments and cancer studies and all of that. So collecting public data and where to reach is important. From where to pick up is one of the very important question, and one should know that. Step three is from data to features. Already very nicely told by we've seen the previous lectures. um, Sorry, guys. Raw data. The raw data is example, if we are working with a FASTA file or we are working with a CSV of expressional counts. So raw data can't be used directly by most AIML models. AIML models actually want us to feed them the numbers, as our machines and AIML data sets are will cannot work on fast and csv directly. They want to learn, and they only can understand the numbers. So what we do here, we do feature engineering. That comes there comes feature engineering into picture. This is a process of converting raw data into numerical formats that we call as features, right? That the model can understand, our algorithms can understand. That we call as a cleaning. We do cleaning, right? You must have done the pre-processings, one hot encoding, label encoding. So all of these are feature engineering that we do before feeding for the validation and test sets. So this is from data to feature is can be of should be your step 3. Okay. uh, Giving you an example for a sequence, convert a DNA sequence into a vector of numerical values representing your kmer counts. This is one of an example. If you're doing your sequence counts, you have to convert representing the kmer counts. For expression data, normalize the raw reads into a matrix of logic transformed values. This is a few of the example for feature engineering, for feature expression data, for your sequence data. You need to stick to all these feature engineering for your data, uh, for bringing that ahead for the analysis. Step 4 is training simple models. All right, what we do is start with simple models, building simple models. Take up the basic inputs like numpy, pandas, ggplot, matplotlib. Don't start with deep learning. Begin with simple interpretable models to establish a baseline. First, establish your baseline. The recommended algorithms would be for classification, use the basic ones: the logistic regression, basic classification regression models, a simple linear model. It will provide you a good baselines. Then you can go ahead with a random forest, which is a powerful ensemble model that works with high dimensional data, and also it is very less prone to overfitting. So you can stick to random forest. For clustering, if at all you want to analyze or hypothesize your endpoints for clustering for unsupervised model, maybe you're working with, so k-means clustering. This is intuitive algorithm for finding groups in your data that will group your data. A few of the tools, the use libraries like scikit-learn and python. Stick to scikit-learn. Very easy to implement these models, easy, and also it will give you end to end a good detailed analysis, right? So you can stick to the basic tools, basic libraries. Train your simple models. Start with that in interpreting and building your models, right? Guys, few of the project ideas that I have picked up is a disease gene annotation. Okay. Whoever is listening to me wants to maybe transition into industry, or maybe starting their projects for academia. Picking up a project, a good project ideas and project is very important. Idea one can be disease gene few of the ideas that is that maybe you can do and is trending is disease gene annotation, drug target prediction, NGS data classifications. Fine. Disease gene annotation. Can you predict, like what can be the hypothesis that can you predict which genes are associated with a specific diseases? Example, a rare genetic disorder maybe by analyzing gene expression data from patients. Um, Talking about drug target predictions is using chemical properties of a drug and structural information of a protein. Can you predict if they can bind together? You can do your docking analysis, your molecular dynamic simulation, interaction analysis. The basic, some of the basic tools were already being shown that you can stick with basic tools like like Primol, Chimera, a discovery studio. You can do that, drug target predictions, disease gene annotations, NGS data classification, basic ones. Can you classify different types of bacteria from a metagenomics sequencing data set? Just go to any of the open data set, maybe pick up something from Kaggle or any of the data open source data sets. Start with your genomic analysis. You can pick up a gut microbiome analysis, and then start building your basic NGS pipelines, NGS data classifications. Use a Jupyter notebook, use R as test set. This is a few of the project ideas you have to pick up, and then follow the research cycle. interpreting and then collection of a data, how to analyze, then evaluate, and then you'll be able to present it with your peer, right? So that is the idea. Conclusion, talking about the next steps, projects are the key to mastery. A project will troubleshoot projects with different with different pipelines. Error solving is crucial, is very crucial. They are where theory becomes your practice. The theory that you've done with your curriculum becomes your practice. Okay, start simple, stay focused, and build on your success. That will be highly encouraged. Share your work, that is important. Use platform like GitHub. Ma'am already told you, GitHub repositories, version controlling. Do that. Always stick to those basic conducts when you're doing your scientific research and analysis, to showcase your code and results. This is the professional research portfolio should look like. This is how you can make your own portfolio, share, and represent, present, and you can even get hired with a good portfolio designs. um, so thank you all for your attention. uh, Over to you, Urmimala ma'am.
Yeah, I guess I'm audible now. Yeah. So thank you so much, Snigdha ma'am, for giving us clear guidance on how to build pipelines and mini projects. That was indeed very motivating and actionable, first of all. And we now know how to put all of this into practice with mini projects, dry lab pipelines, and project ideas that anyone can start, whether you're a student, a researcher, or even a working professional, right? So thank you once again, all our speakers, for the insightful session. So now here's something very important. I'm sure many of you are wondering, where do I actually start learning and applying this? Tell me honestly, you are wondering or not? Are you thinking that where do we learn all this? Yes, how many of you are wondering? Correct? That's right. That's right. All right. So the truth is, figuring it out all alone can take months, right? Exactly the platform, right, Satyajit? Yes. So you are not alone in this. Yes, if you have to do all by yourself, search the material, search the platforms, it can be really troublesome for you, and you need all at one place. So that's where Biotechnica's AIML internship in biology and bioinformatics comes in. So you learn in bioinformatics and computational biology. The best part is it is an industrial training program, right? So what exactly does that mean? It's designed as a plug and play system, you can say, for students and researchers, right? Where you get guided mentorship, you get to do your own project, and that to ready to use projects, you can get your own publications. Our principal investigators will be assigned from our side for your projects, okay? And online, offline project work is also available, whichever you want to avail. And most importantly, the coursework before the internship, so that you know what exactly to do in the project. Right now, you if you are a beginner, so many people said, right? So Vik Leaf is asking what should be the beginner step. So that's what. First you learn the things and then you apply the rest of the stuff, right? So just to make it simple, and for beginner level, we have structured frameworks for our courses. You shouldn't delay in this because our course is starting on 29th August. Yes, this Friday it is starting. You get hands-on training, you get to publish your papers, and as I mentioned, you have a structured curriculum to begin with. Okay, so do not delay. The registration link has been given below. You can click on it. If you're interested, you can also call our team to get more details on this course, right? And to find out that how it is going to benefit your career. And then you can just, we will see it in in our classes, right? All right. So this you're serious about making AIML a part of your research career, then this is the best place to begin. Do not think twice. Okay. Yeah. So we will now open the floor for your questions. Many of you are eagerly waiting for this. Okay, so feel free to drop them in the chat. But since we have crossed little bit of our time limit, so we will take some limited questions. Okay, so let's see. We will take up the questions which was already previously posted by our students. So we have one question from Mr. Sandeep Barik. This is directed to Dr. Anu ma'am. What path would you recommend for someone with a bioinformatics background to grow into product management in life sciences? Dr. Anuprabha ma'am, we request you to kindly reply this.
Yes. Hi, Sandeep. I don't know, but maybe, okay. So first of all, you know, you need to figure out what is your area of interest. Or life figures out for you when you get your first job. So generally, you figure out what is your area of interest. If it is so most of the companies that are there are either working in the drug discovery industry or they are into genomics. Most of the automations and product based companies in are towards genomics currently, as much as I know, because I have mostly been in genomics. So in case you want to do that, uh, I think a basic understanding, as I said, a problem-solving attitude, a troubleshooting, attitude, finding a problem, why it might have occurred. I think those are thought processes. A product manager would have. What is the demand in the market? How to go about it? How will I solve that problem? And delineate problems into various aspects. I think that is something. If you have that mindset, if you have that interest, I would really urge you to start basic. At an analyst role, do it yourself. Get your hands dirty as much as possible. Just have 2 to 3 years dedicated to just polishing everything, and then I think is the right time when you have a breadth of knowledge of getting into even the starting of any product role. So yeah, so that is the journey that there is. I hope I
Definitely,
answered
ma'am.
your question.
Yeah, absolutely, 100%. That was the actual practical answer that you have given to our students. So thank you for that. Yeah, so I would like to take up one more question from Satyajit Das. Should we completely rely on the AI ML for our research work? Dr. Neeraj Kumar, would you like to take up this question, please? Should we completely rely on AI ML for our research work?
Of course, ma'am. So thank you for your question, Satyajit. So I do not agree with this statement, because AIML comes in action whenever you have the foundation and you have the query, right? So what you want to predict, and if you have research question, and why you want to implement AI as well as ML algorithms, right? So a clear definition of question, as well as what type of data you have and how to handle that data. Okay, so for all these to answer all these questions, first of all you need to put your biological question there. Okay, so what is what you want to achieve? Okay, why you need to implement AI and ML? Then you need to go to AI ML. But before that, you have to have the basics of the problem, and you need to understand the problem. Sometimes AI ML is not required at all. Sometimes the basic programming skills, as well as the basic handling of data, just serve the purpose of the problem, right? So AIML is a technique. It is just a tool, like you are just going through other experiments as well as the approaches. Similarly, AIML is just a technique. So that's it.
Thank you, sir, for that insight. Okay, so one more question from Mr. Abdul Mubarak. In bioinformatics career, MSc is enough, or should we pursue PhD? Is it necessary or not? Snigdha Tiwari, ma'am, would you like to take this up, please? Ma'am, you're on mute. Um, Ma'am, you're still on mute.
Wait. Can you hear
Okay.
me, ma'am?
Yeah, yeah, now it's fine. Now it's fine.
Okay, okay, fine, yeah. Okay. So Abdul, talking about your question, I think what is your niche matters. In many cases, masters is, maybe enough. Many time, a PhD is not always enough. So what is your niche? What typical roles you're wanting to get in, that matters. Say for example, you want to get in with academia, big tech, research labs, maybe a core R&D. Then PhD might be a necessity, right? For a research scientist or a professor, or something like that. If at all you start to work with any of the industry, you're transitioning into it, applied R&Ds and all of those areas, all of those lines, then maybe MSc and MS does the job for you. Of course, with certain skills, certain pipeline developments and everything that you must have. A few of the typical roles, maybe after MSc, some specialist, a data scientist and all of that. So you can go ahead with that, why not? So what is your niche and what roles you want to achieve? What is your vision matters. So I hope it
Yeah,
answered your question.
yeah, definitely, definitely. Thank you, Snigdha ma'am. And basically, you have to explore a lot of things. Now, these days, PhD is not that much necessary. If you know that where you want to explore, as ma'am said, and try different things, different areas, different certification course, different internships, right? So I have one more question from the same person, Abdul Mubarak. But this question, I think many of you would be having. So I would request Dr. Anuprabha ma'am to kindly take this up. What are the top companies hiring for bioinformatics in India as well as abroad? Can we get full-time remote job in bioinformatics? Which career will not get replaced by AI? Yeah.
Uh, okay. Hi, Abdul. Careers that are not going to be replaced by AI are thinkers. They are not going to be replaced. Uh, Identifying a problem in the market, getting the right solution and the easiest solution. Most of the times, I think when I take interviews of most of the students that are there who are freshers, they emphasize so much on complexity, but the entire industry is built upon people who make the most simplistic solutions to the most difficult problems. If you have that mindset, I think that is absolutely necessary. uh, Again, talking about top companies that are hiring for bioinformatics in India, I think there are a lot of jobs out there, and specifically, I think in case you want to explore, as you know, in the previous, the answer that Dr. Snigdha also tried giving, and others, that if you want to really know what industry would be like, your day to day would be like, join internships. Those are the best. They have no risk. You have, a toll of industry people just sitting around doing their work. Go and observe everyone. Just go there, do that, 3 month, 2 month, 4 month, whatever you can get your hands on, and go and do that internship, and it will open horizons for you. What is your interest? Most of the people, I think, struggle with, I don't know what industry I want to go. Should I learn 15,000 tools? No, you don't need to learn 15,000 tools. You need to know basics. As I said, Python is something that is very important in industry, because a lot of all of the pipelines, all of the things that I know of are done in Python. So that is something, it is very basically Python, Bash, R possibly. So, but yeah, if you know the basics really well, you know what to do about it. I think that is enough to get around any interview in India, of course. Talking about abroad, so I have been only working remote. So it has been, I do not know how many years that I have not gone to office. I am working remote. I have my entire setup done here for remote, and my company right now, the headquarters is in San Francisco, and the people I work with are in Tel Aviv, Israel. So Yeah, and I'm talking to you guys. You can work remote. There are a lot of remote options, but remote opportunities, I think, is you will be able to crack only if you are in the advanced uh, uh, place in your career, slightly intermediary probably. But I would just urge everyone, if you want to, if you are a fresher, look for a good startup that has a good growth journey for the last 2 to 3 years, join it. Forget you are a PhD, forget you are a master's, forget you are a bachelor's for that matter. If you can get in, that's it. You can decide what you want to do once you look at the day to day. If you don't get a chance to do that, you will never know from your books what is there to, waiting for you at the other end. So, yeah,
So
I
true,
hope
so
it
true.
answers
Yes,
your question.
yes, yes, ma'am. As you previously said, you have to get your hands dirty. Then only you get to know what it is all about. Yeah, okay. So one more question from Sandeep Barik. I think this question also is quite important. In the context of healthcare and precision medicine, what specific roles or responsibilities does a bioinformatician handle? For example, in genomics, diagnostics, or patient care. Dr. Neeraj Kumar, would you like to throw some light on this question very briefly?
Yes, ma'am. Yes. So, being a bioinformatician, you have various roles in genomics, patient handling, as well as the diagnostics. So, you can work as a data curator or data analyst. So, while working as a data curator or data analyst, you can work over various data sets, how to handle those data set, how to curate those data set, how to make a sense out of those data sets. So, you can provide them publicly. Okay, so while handling a patient, so you can design various plans as well as algorithms or various approaches with which you can handle the patients more carefully, and you can design the prescriptions very well. Okay, and being in diagnostics, so you can develop various algorithms. They can be bioinformatics approaches as well as the AI/ML approaches to find out various biomarkers within the lab tests. So yeah, these are the brief overview regarding those rules.
Thank you, Dr. Neeraj sir. So this another question is from Aniket. Do I have to learn all the mentioned tools if I want to pursue career in computational biology? I'm a core biology student, but this is a kind of diversion from the core biology areas. Snigdha ma'am, any insights on this? Very quickly. Ma'am, you're on mute.
So Aniket, I think ma'am has already told you about that. Learning all the tools is not important. Your basics should be very good. Say for example, learning all the languages doesn't matter. You have to master one language. A lot of questions, even I get through that, should I learn all the languages? I should know all the tools and languages. My answer would, I will highly encourage you to start with basics. Get a good get master of any one language. Say for example, you're good with Python, you can seamlessly shift to any of the languages. You can know the libraries, you can learn anywhere, you can pick up anything anywhere, right? Because a specific language, few of the tools of your niche are well. So I think knowing everything doesn't matter. What you know, the best matters. Ma'am already told that. I've just extended that, so I hope that answered the question, Aniket.
Yeah, thank you. Thank you so much, ma'am. So I will take one last question, and this is from Skynet AMVs. Uh, I would like Dr. Anuprabha ma'am to kindly answer this one. When defining a problem, actually, I think it's directed to you only, ma'am. When defining a problem, sometimes it feels overwhelming to narrow down a big biological question into a precise, researchable one. Are there any strategies you use to make that process easy?
Okay, so hi, I don't think this is your name, so I would like, hi, Skynet. I don't know. Yeah, uh, okay. I do understand why you would want to, why you would feel like, something like this can be overwhelming, because when you want to break down in a problem into small steps, it does take a lot of, horizontal knowledge, which, if you have done your masters, or if you know biology well, and if you have any understanding of what I want to accomplish in each step, it would help a lot. For example, if I say, I from this data, I would want to find what are my cancer defining mutations, for example. Okay, because I am trying to, maybe do something about it. Now I have data. What kind of data I have? Now, if I have a data to actually do something about it, I will need to, sort of transform the data, do some processing on it, normalize it probably, okay, do some sort of feature optimization, so on and so forth, right? So, thinking, so what has helped me the most is looking at the outcome that I need and finding the shortest route that is there. And whatever steps I could think of, writing it down at one place, it's like preparing a pseudocode in your mind. So, for any coding that you need to do, I don't think the language is as important as much as the pseudocode. How you think about it, what is the logical steps you take. Like, if I want to do something, first I need to just input the data. So you start with, inputting the data, and now that you have the data, you cannot do any other operation because there are 15 steps to it, right? Because you see, the format is not right, or other things are not right, and you need to just prepare that pseudocode for yourself. And once you prepare that pseudocode for yourself, you can, for each of the problems that you have come across, you can just go and Google. Everybody has to Google. You cannot remember everything. Just go and Google. This generation is of AI. You can go and ask Chat GPT, Copilot, DeepSeek, whatever suits your fancy. Go and ask it. If you don't know how to break this down, you go to Copilot and ask it. You know what? Break this down into these many things. But what you need to do, no AI will work for you if you do not know how to frame the problem. No AI. The person who can use the best, can use the AI to the best of their capabilities, is the person who knows how to explain the problem. And to explain the problem, you need to understand it really, really well. So if you know how to explain the problem, then you can ask your chat GPT, your whatever it is, those problems, it will break it down for each. You can go and see what are the tools that are there. You can, choose, no, 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 give me the most beginner friendly tool. Do it yourself. See the outcome. And then you will have some sense of confidence that, I'm going on the right track. Don't need to just, I have not met any person in all of the like 10 years of of industry. I have not met any person who has said, you know what, I know this language completely. It has never happened. So because there is a discovery every day, so you can never know it. It's okay to not know it, but it's okay to know why you don't know it, and it's fine. But you need to understand what you're trying to solve. If you don't know the objective of the problem, so one instance that I do generally in my interviews, the first thing I say, you have done this project, why did you do that? And 90% people haven't been able to answer it. So you just need to know that. So just try to understand the problem and then look for solutions. Do not start from the other end. I have to fit ML into everything. It will not fit. I have to fit AI into everything. It will not fit. You will just do something so many times wrongly. You will realize, you know what? I think there is a better ML model out there. I will use that. So that's how it is.
Yeah. Thank you, ma'am, for the elaborate explanation. I'm pretty sure everyone was very fascinated with this answer. So, with that, we come to an end of this Q&A session. And many of you are asking, like, how to start a project and what you need to know before a project works happens. So that's what you need to go into the depth. Okay, start with the basics. Join the internship. Somebody has asked how to where to start reading about coding from scratch. How to learn coding from scratch. Okay, so the answer is in front of you, as our speakers also said today. Join the internship. Once again, a reminder. We have our AIML in Bioinformatics and Computational Biology, which is starting on 29th of August. All your answers, how to start project, what you need to learn, everything. This is a one-stop answer for you. Okay. So with that, as we close, I would once again like to sincerely thank all our speakers, Dr. Neeraj Kumar, Dr. Anuprabha ma'am, and Ms. Snigdha Tiwari ma'am for sharing their time, expertise, and valuable insights with us today. And of course, a big thank you to all our participants for being an engaging audience, a super supportive audience. Right? So remember, the future of biology is computational, and the future of research belongs to those who learn AIML today. With that, we conclude this session. Thank you all for joining and have a wonderful evening ahead. Goodbye.
Thank you.
Thank you. Thank you, everyone.